for this morning's presentation. And we welcome to Seba another new addition who joined at the same time as Dr. Parisa on 31st December, Dr. Nurul Huda Muhammad Iqbal. And she's actually completed her PhD in together, I think, with Dr. Suba's group, who she continues to work with. And her study in the PhD was on diversity of rats in urban areas to understand the complexity within the species and population structure in relation to the zoonotic disease, specifically leptospirosis, which I think we've all seen in the media as being a sort of a problem that crops up from time to time in Malaysia, and why we all wash our drink cans and are very careful with our hygiene when eating outside. So her current research focus is on the karyoological and molecular characterization of marcia, sea bass, and grouper fishes, so from rats to fish. And being a young researcher, she's looking forward to more collaboration opportunities in various research fields as part of her ongoing career development. So again, this is another opportunity for you to get to know one of our postdocs and to follow up with them. So to give her more time to speak, I will stop now and invite Dr. Nurul Huda to present her seminar to us. Thank you. You're on mute, I think. Nurul, turn on your microphone.
Sorry. Okay, thank you, Prof. Jenny. Assalamualaikum warahmatullahi wabarakatuh, and a very good morning to all. I'm Nurul Huda binti Muhammad Iqbal, currently a postdoc under Terra Aqua Group in Sabah. I'm truly honored to be invited as one of the speakers for today's event. Just now, both speakers talk about plants, but now I want to talk about animals, which is part of my PhD study. So without further delays, let's start this session with my presentation entitled Species Diversity of Rattus, Rodentia muridae on the Malaysian Peninsula, inferred from complete mitochondrial cytochrome B gene sequences. So this is a basic outline of my presentation. So as for introduction, genus Rattus, which is mainly known as a major pest in agricultural farm, is a species complex which the boundaries between them is often unclear. There are many synonyms for each species. Usually the identification based only on morphological features and size are really confusing. The taxonomic status always been changed by different authors, where several authors might agree on the full species status and some might agree on the subspecies level. So this genus Rattus is poorly understood from a taxonomic and evolutionary standpoint. So it is really important to know what is the current status of Rattus in Peninsular Malaysia. So here are the several references used in this study to reveal the taxonomic status of the rats in Peninsular Malaysia morphologically. So the latest references, which is by Francis 2008, he discovered 6 species of rats from Peninsular Malaysia within the genus Rattus, which are the Rattus, Rattus exulans, Rattus argentiventer, Rattus norvegicus, Rattus tiomanicus, and additional of Rattus annandale. Actually, when we look at this morphology of these 6 species, is actually almost the same in the field. So, however, Camargo Sanchez et al. 2017, he corrected the Rattus annandale into a new genus, which is the Sundamis annandale. So now leaving behind 5 species within genus Rattus in Peninsular Malaysia. So for our study, we used our background study is actually following Aplin et al. To fill in their gaps. So their study is Aplin studies is actually aims to resolve the taxonomy of the black rats based on the evolutionary history and geographic pattern of diversification. So from their findings, Aplin found out there are 4 lineage of black rats, which is Rattus complex lineage 1 to lineage 4, which is the lineage 1, 3 and 4 is actually representing the Rattus, and lineage 2 is representing the Rattus tanezumi species. So and they also found found out two other recognized species, which is Rattus baluensis and Rattus tiomanicus in lineage 6, while the Rattus losea in lineage 5. So this figure 1 shows the geographic distribution of the 6 lineage of the Rattus complex by Aplin et al. As we can see here, as we can see here, the lineage 1 is actually as we can see here, the lineage 1 is actually representing the Rattus sensu stricto, which is mainly found in India. Rattus lineage 3, which is found in Himalayas of Pakistan and Nepal here. And Rattus tanezumi, which is lineage 2, is found in India, Myanmar, Laos, Vietnam, and China. Lineage 4 is found in Laos, Thailand, and Vietnam. And they also found out that there's the Rattus complex lineage 5 occurred in Laos and Thailand, while lineage 6 is occurred in Java and Borneo. So figure 2 showing the global distribution of sampling localities by Aplin. However, we can see that the samples from Peninsular Malaysia is absent from their study. So we intend to study our genus Rattus in Peninsular Malaysia to further validate the status. So why we studied this is because the taxonomic status of several Rattus species from Peninsular Malaysia is still unclear. Their genetic relationships also remains backdated. The identification of skull and dentition characteristics remain difficult due to morphological divergence within the Rattus species. And finally, the molecular records of Rattus species from Peninsular Malaysia were very little. So from the background study, several research questions arise. First, how many Rattus species present in Peninsular Malaysia, and do Rattus sensu stricto is one of them? Second, in which lineage do Rattus from Peninsular Malaysia belongs to? And third, which species found in Malay Peninsula, whether they are native or invasive? Here we would like to know. So our aims is to resolve the molecular complexities within genus Rattus from Peninsular Malaysia using cytochrome B gene. Second is to examine the phylogenetic position of the Rattus species complex from Peninsular Malaysia through molecular data. And third, to determine the locality of the species found, whether it is native or invasive in the Malay Peninsula. So for methodology, first we did our samples collection. Altogether, we collected 35 individuals from the genus Rattus from 6 main states in Peninsular Malaysia, which is Perlis, Kedah, Penang, Kuala Lumpur, Melaka, and Johor. So, as a commensal rats, we targeted 3 main areas, which is a fresh market, seaside, and the housing area to trap this rat. We use the cage trap. The cage trap breeds with banana in the during the trapping session. So we did our molecular study when at first we sacrificed the rats and take out the tissue samples, and we isolated the DNA, and then further doing the PCR amplification, which targets the cytochrome B gene, which is the mitochondrial from the mitochondrial organelles, and then followed by the gel electrophoresis, and finally by doing DNA sequencing. So for the molecular analysis, the DNA sequences, the edited DNA sequences were blasted through the GenBank database, and we did the sequence alignment through cluster W in Mega software. Then we did the phylogenetic analysis and construct the neighbor-joining tree using Mega also, and did the molecular dating to know the evolutionary time point using the Beauty and Beast software. Then finally, we did the minimum spanning network through the DNA, SP, and network software. So now we move on to the results. From the 35 individuals collected in Peninsular Malaysia, the molecular taxonomy of Rattus shows that there are 6 species occurred in Peninsular Malaysia, which are Rattus tanezumi, Rattus tiomanicus, Rattus lineage 4, Rattus argentiventer, Rattus exulans, and Rattus norvegicus. So as you can see here, the green color is actually the Rattus lineage 1, which is sensu stricto, which is thought to originated in India. And this one, the red color is lineage 3, which is happen to inhabits in Himalaya, is actually absent in Peninsular Malaysia. So this figure 4 shows the minimum spanning network of and then we can see this is a big interesting result in which we can see that the Rattus lineage 4 found in Peninsular Malaysia is actually in a different subclades from the one studies found by Aplin, which is in the light blue color. So this minimum spanning network of genus, this figure shows the minimum spanning network of the genus Rattus haplogroup. So I would like to just focus on the blue color group, which is the Rattus lineage 4 found in Southeast Asia. So the light blue color is the samples collected by Aplin. And here we can see that the samples collected by Aplin is actually from the samples of north of Isthmus of Kra, which is from Laos, Cambodia, Vietnam, and together with the samples from Indonesia and Sri Lanka. And our samples collected is actually Peninsular Malaysia, which is located in the south of Isthmus of Kra, is actually grouped together with the southern Thailand, which is also in south of Isthmus of Kra, and together with the Philippine samples. So the genetic distance data further validate the separation of the lineage 4 found in Southeast Asia, which is the number 7 here. This taxon is actually studied by Aplin, and the number 8 is actually our samples from Peninsular Malaysia. And we can see that the divergence of 2.1% is discovered between these two groups. And according to Bradley and Baker, the genetic distance of 2 to 5% from the cytochrome B gene is actually can further tell us that they might be a subspecies, they can be considered as a subspecies level within this range of genetic divergence. And also we can see here the Rattus belvensis, which is number 5, and Rattus tiomanicus, which is number 6. They are having just a less number of value of genetic distance, which is 1.9, which shows that actually both of them are a valid different species. However, their genetic distance is very small. Okay, this figure 5 shows the minimum spanning network of Rattus species haplogroup with specific geographic origins of the haplotype. So from here we can see that to further know our samples, which samples is invasive or native to Peninsular Malaysia, we did do the sequence comparison analysis from this result showing that the Rattus tanezumi, Rattus norvegicus, and Rattus exulans is actually invasive in Malay Peninsula, while the Rattus lineage 4, Rattus argentiventer, and Rattus tiomanicus is a native species to Malay Peninsula. So this is the results of this analysis of the Rattus species. So as we can see here, all the diversification within each of the species of genus Rattus estimated to have commenced around 0.21 to 0.233, which is during the Pleistocene. So during Pleistocene, the most of the mainland and most of the earth land is covered by the ice, and the land is actually connected together. But due to sea level rises in Holocene, the land separated as is happening today. So it makes the animals actually difficult to move, to migrate. So as for discussion, the molecular complexities do exist in genus Rattus. As we can see, there are distant relationship between the Southeast Asian Rattus, which is lineage 4, towards the Rattus sensu stricto, which is in lineage 1, and Himalayan Rattus, which is lineage 3. Aplin et al. Studied that they might the factors of the complexities might be due to the chromosomal variation, which they found out that the Rattus lineage 1 do have 38 chromosome number, Rattus lineage 3 do have 42 chromosome number, and one of the samples they collected in Sri Lanka, which is belong to lineage 4, do have 40 chromosome number. However, even our samples is actually in lineage 4, but we cannot confirm our chromosome numbers since we didn't do the cytogenetic study. So the complexities also might be due to the geographical barrier, which the sea level rise caused the isolation of the three lineage of the Rattus. Isthmus of Kra, which is the narrow neck of the which connected the Malay Peninsula with the mainland Asia, may have influenced a postulate disjunction to represent geographical patterns of population subdivision within the Rattus lineage 4, as we can see that the north of Isthmus of Kra and south of Isthmus of Kra having are separated in different subclades. So the close relationships found out between the several species between Indo-Chinese and Sundalian population might be due to the migration across the land bridge during the Pleistocene. So based on molecular evidence, there are multiple species within what has historically been identified as Rattus. And from this study also, we found out that there are the occurrence of Rattus tanezumi in Peninsular Malaysia together with Rattus as a sympatric species. So as for conclusion, there are at least 6 species of Rattus discovered in Peninsular Malaysia throughout this study. The Rattus from Peninsular Malaysia belongs to lineage 4, and we need further to confirm their status further, and it is not the Rattus sensu stricto. And we also found out that there are 3 native species found in Peninsular Malaysia, which is Rattus lineage 4, Rattus tiomanicus, and Rattus argentiventer, while the another 3 of Rattus tanezumi, Rattus exulans, and Rattus norvegicus is as an invasive species in Malay Peninsula. So as for recommendation, the Southeast Asian Rattus lineage 4 should be treated as a discrete genetic entities in order for the management of the species, as most of us know that the Rattus is actually classified as a pest. So however, for the management, we need to know the genetic entities at first. So there are also suggestions in naming the black rats. So it is also important to recognize the genetic diversity between the populations of Rattus to conserve the unique evolutionary lineage of the species. Broader geographical sampling with the integration of morphology and genetic study using various molecular markers might help to resolve the relationship of the Rattus group across the Malayan region. So I would like to acknowledge each and everyone in the list for helping me during the study, together with Seba who helped me also. And these are some references used in this study, and that's all. Thank you.
Thank you, Nur Huda. Very interesting, and probably very important too, because we know that the rats are carriers of diseases, and we've seen emerging pathogens like COVID, the havoc it can cause. So I think it's important we know more about this species so that we will be able to, well, as you say, control the animal itself as the pest, but also maybe there's different potential of different diseases emerging. And there are some theories even for COVID that it might have come from Southeast Asia rather than China, although, there's no clarity on that at all, but it's a worrying suggestion that's coming out. I open this for questions, and we should mention that now Nur Huda is also working on fish. So although she's presented work to us about rats, she's also quite knowledgeable about fish and working with the fish group and genetic populations in various Malaysian fish. So any questions? It can be about the talk or about also you could ask her about what she's doing now.
Yes, can I have one?
Yes.
Fikri, right?
Yeah, hi. Sorry, Prof?
Yeah, please go ahead, Dr. Fikri.
Yes, okay. I have just a question regarding the invasive species. So you said that several of the species, the rat species, are actually invasive. I'm just wondering, how did you came to that conclusion? And yeah, based on the analysis that you did.
Okay, as we can see there, can I show the slide? Yeah. As we can see here, actually it is stated that Rattus tanezumi actually originated from Japan. And we can see here, when we do the sequence comparison analysis from the gene bank, actually there are many species, there are many individuals caught in various, from the Japan, and another species I had right here, I had right here the origins of the species, of the individuals. And it is stated that the Malaysian is one of the country which the rats migrates from Japan. It's also same for the Rattus norvegicus, which actually is originated in Norway, and this Rattus exulans is actually originated from French Polynesia, and it all migrated, and it is invasive here. But compared to the native species, which is the lineage 4, we can see that it is all just found in Southeast Asia, and we can see that actually our Malay Peninsula samples also, we can say that it is actually uh, originated in Malay Peninsula, which is and all the three, Rattus lineage 4, argentiventer, and Rattus tiomanicus, where they cannot be found outside Malay Peninsula.
I see. Okay, I think, yeah. All right. I sort of get what you mean. And another question is, I saw that the population, the sampling sites that you did for the study, you focus on the west coast of the peninsula. What happened to any comment on why the east coast was not covered as well?
Actually, I did study. My study actually focused on throughout the Peninsula Malaysia, include the East Coast area. But for this phylogenetic study for the molecular complexities, actually I includes all the individuals, which I have 130 sequences, but
I see.
the genetic is not different at all. Actually, I just pick up some of the samples because it's almost the same.
I see. But you did cover like the whole
Yeah, actually,
peninsula.
because I did the prevalence on the leptospirosis also.
I see. All right. Thank you so much.
Okay, thank you.
I wonder why the Malaysian rat doesn't go out. You see that a lot of other rats are coming in, and I think it's interesting that I think rats have long been known to move around on ships, and Malaysia, of course, has been trading by sea for a very long time. So that's given plenty of time for shipping coming from various other ports to bring in rats from all over the place. But the Malaysian rats only in Malaysia. They don't.
No,
They don't
no, actually, now it's they stated that this rats is actually haven't found in other country, and I mean outside Southeast Asia. Maybe there are they haven't studied about it. Maybe
yeah. Perhaps it's a lack
no
of
reports.
information rather
Yeah,
than a lack of
yeah.
it happening. Yeah, that's always an important point in science, right? Just because it hasn't been seen doesn't mean it isn't there if nobody looked.
Yeah, it hasn't been found yet.
Yeah. So
Same goes with China, because you mentioned about the sea trade. So like the trade extend from Europe all the way to China. So I guess Malaysia is like in the middle point of the whole trade system. So I guess the distribution of the rats can actually extend all the way to China. Maybe there are the Malaysian native types in China as well.
Yeah.
Just a comment there. Here.
Yeah, and I think it's also very important, again, going back to because you're mentioning like the zoonotic diseases and of course, how effective are modern day shipping management at preventing, the movement of pests like rats. Because I think nowadays that probably should not be happening anymore, right? But I'm not sure. I remember that another contamination that I was not aware of is by the bilge water. So we're getting all sorts of exotic algae and seawater problems from ships moving around. But I guess by now we probably shouldn't be getting new rats into the country. Hopefully not. Do we have questions or comments from anyone else? I know we have overrun on time, and I thank everybody for staying with us. We still have 25 people, which I think what we started out with, so that's really good. just before we go, if you can still ask questions if anybody wants to, but just a reminder that I think on the side in the chat box you can see the link to the Google Forms. Please just take a minute or two to finish the survey for us. That would be really helpful and most appreciated. And of course, this has been recorded, so we will be able to make this available to a wider audience if the speakers agree to us doing so that they can be known. Call for questions?
Maybe I can ask just a general question, a very interesting talk.
Thank you.
Since we're talking about the prevalence of the leptospirosis, is there a preference of species or lineage in terms of the prevalence of the leptospirosis in the Rattus?
Okay, actually in my PhD thesis, my main focus is actually on the Rattus lineage and because it is the taxonomic status is confusing, right? So I did the prevalence study only on Rattus, but I also tested on some samples species like Rattus tanezumi, but I haven't reported. I didn't publish it, but I also did lab work and tested that Rattus tanezumi, Rattus tiomanicus also is actually carrying leptospirosis. But for Rattus, I just discovered the leptospirosis prevalence in states. I mean, I just saying that like Rattus collected from fresh market is having more prevalence carrying leptospirosis compared to the seaside area. So it means that the leptospirosis is actually a bit danger for us as we are all going to market and it's we need to be caution.
How about in terms of East Coast, West Coast?
East Coast, West Coast. There are also I also did the prevalence, but the prevalence is not as much as I got from Kuala Lumpur. Maybe there's bias also, as from Kuala Lumpur I got a lot of rats. I mean many number of rats. But in East Coast, if I'm not mistaken, from Terengganu is only 12 and Kelantan is only 8, something like that. And the prevalence might be less because of the bias.
Okay, thank you.
Okay.